Hi everyone, we're going to have a look now at bacterial transformation, so an extension of DNA manipulation using the techniques that we've discussed previously. So there's three main steps involving bacterial transformation. We need to get the gene that we want in the plasmid, so usually means we need to cut it out of something, and to cut we use our restriction enzymes. We need to get the plasmid into a bacteria, which means we need to ligate or use our DNA ligase. And then we need to get the bacteria to replicate and make lots of copies of that gene. Here's a summary of this process. So we've got a human cell, and from that we've extracted an insulin gene. That insulin gene has been inserted into a plasmid that's been extracted from a bacterial cell. When we insert the gene into the plasmid, it becomes what we call a recombinant plasmid, a plasmid that's had a gene inserted into it. That bacteria that plasmid, I should say, is then put into a bacteria. That bacteria is then provided with nutrients and agar to grow in a culture. That bacteria is then extracted, and we're able to transform or put that into a person to give them insulin. We'll look at how we transport DNA into cells. So very often DNA needs to be moved from one organism to another. We can't simply inject a cell with one gene because it will quickly degrade. So we need special carriers known as vectors, and they're used to transport DNA into a bacterial cell. Usually this vector is a plasmid, so a circular piece of DNA that's extracted from a bacteria. Plasmids are small pieces of circular DNA. They can self-replicate easily, starting at a point called the origin. They can easily have genes added to them using restriction enzymes, and many genes that make it easy for them to see if the transformation has worked. So plasmids are separate pieces of circular DNA from the main chromosome in a bacteria, and they replicate by themselves independently of that main chromosome. Special features of these plasmids: they've got selectable gene markers, so a gene that governs antibiotic resistance, specifically to an antibiotic called ampicillin. This makes it easy to find bacteria that have successfully taken up the plasmid. They've got multiple cloning sites or a polylinker, and this is a region of the plasmid DNA that contains recognition sites for lots of restriction enzymes that produce sticky ends. So this makes it really easy for us to be able to insert genes into these plasmids. So this is an example of a plasmid, and it shows the ampicillin gene. So this is the one that's responsible for antibiotic resistance. And then it shows how it can be cut by lots and lots of different restriction enzymes, which makes it really useful. So depending on what recognition site we need, we can select what restriction enzyme we use to cut to be able to insert the gene that we need to get in there. This diagram here is showing us bacteria that are killed off, are not resistant to antibiotics, and as a result, they're killed off. a plasmid survives because plasmids have got genes in them which are resistant to antibiotics. So if we've put a gene into a plasmid and then we put it onto our culture to grow, and then we put antibiotics on it, only the plasmids that have got that introduced gene are going to survive. So then we know which ones we've got with our that are transformed or that have had the new gene inserted into them. Let's have a look at how we make a recombinant plasmid. So the DNA of the plasmid is cut at one point using our specific restriction enzyme that creates sticky ends. This changes the form of the plasmid from circular to linear. So once we cut at our recognition site, we go from a circle to a linear piece of DNA. The foreign DNA fragments are prepared using the same cutting enzyme so that we've got our complementary sticky ends. So that it has those complementary sticky ends. So foreign DNA and the plasmid are mixed and the sticky ends pair. The plasmid with the foreign DNA is called a recombinant plasmid. So here we've got our plasmid. Here is our piece of foreign DNA. When that foreign DNA has been inserted and stuck to those complementary sticky ends, it becomes what we know as a recombinant plasmid. Recombinant means it has the gene stuck in it. The joining enzyme ligase is added, and that's what permanently joins through the process of completing those covalent bonds between the bases. Getting the plasmid into the bacteria. So bacterial transformation. So a plasmid is the carrier or the vector to introduce a new gene into a bacterial cell. So when foreign DNA is directly taken up from an external source by a bacterial cell, those bacterial cells are said to be transformed. So transformed bacteria have got a new gene inserted. Getting plasmids into bacteria. It's quite difficult to force bacteria to take up plasmids, so the odds can be improved by doing a couple things. What we call electrification, so shocking the bacteria with electric fields, and heat shock, which is where we place ice cold solution and then transfer them to a 42-degree solution for a short time. Both of these methods aim to increase the fluidity or the permeability of the plasma membrane, which increases the likelihood of the plasmid entering. This is just a diagram which is outlining the steps in that heat shock process. Why is this useful? So we can inject any gene into bacteria. So for example, we can get the GFP, which is a green fluorescent protein from a jellyfish, insert that into a bacteria and make it glow. So because bacteria multiply quickly, it means we can make lots and lots and lots of copies of that gene. And that's often what we want to do, is make those lots of copies of the gene. Making multiple copies. Once the recombinant plasmid is in the bacterial cell, multiple copies of the gene can be made in the process called gene cloning. So this occurs because as the bacteria replicate, it also replicates the gene that's in the plasmid. So therefore, we're continually making lots of copies of that new gene. So we're cloning it basically. This diagram here just outlines the steps in this bacterial transformation and gene cloning process. Let's have a look at some questions. So this question here says bacteria can be transformed with an artificial insulin gene and culture to make insulin in commercial quantities. The steps taken to produce the genetically engineered insulin are summarized below. The order of the steps has been mixed up. Okay, so basically we're looking for the correct sequence of steps involved in this process. Okay, so to answer this question, and I know the solution is just there in front of you, but to answer this question, we basically need to order those steps. So the first thing that happens is that we make an artificial gene by adding a base in a gene synthesizer. So we're making the gene that we want to insert. The second thing that happens is that we insert the artificial gene into the cut plasmid. Following that, we add the recombinant plasmid, the plasmid that's had the gene inserted, to bacteria, and heat to enhance the uptake of this plasmid. We then identify successfully transformed bacteria. The culture transformed bacteria are harvested, and then we purify the insulin. Then we devise a nucleotide sequence by working backwards to form an amino acid sequence. Then we isolate the insulin from human cells and determine its amino acid sequence.